So Marcus, hello. Could you introduce yourself?
Yeah, so my name is Marcus Horn from Leibniz University Hanover. I'm there at the Institute of Microbiology, and my main area of research is basically microbial ecology.
Okay, so what does it mean, microbial ecology?
So microbial ecology is a field of research where we deal with the interactions of microbes with each other. So consider, for example, a gram of soil. You might have 1 billion microbes in 1 gram of soil alone. So microbes are really abundant in the environment. You have maybe 1,000 to 10,000 species in there, and they're not living secluded from each other, but they are interacting and forming a very complex community. These communities have activities, they perform ecosystem services, processes we are interested in, and we like to have a look and get insights into the interactions between different microbes and the communities and how they develop over time, most of the time with respect to some processes we are interested in.
So we can use these interactions, for our purposes.
And we try to use these interactions for example, having the removal done of certain pollutants in the environment.
What kind of pollutants, for example?
Pollutants might represent pesticides in our case, might also represent the so-called micropollutants, pharmaceutical residues that end up in the wastewater and lastly in the streams and rivers. So we try to use the sediment communities in this particular case to get rid of the pharmaceuticals that remain in the streams and rivers after receiving the discharge from communal wastewater treatment plants, for example.
So what is your role in NYMPHE project?
And this really is related to the NYMPHE project now, because we try to enrich and isolate organisms from the sediments of rivers and streams from the so-called hyporheic zone, which is a very active part of the sediment where oxygen-containing water penetrates the sediment and a lot of microbially catalyzed reactions are going on, and where we find also a lot of different microbes in there. We try to get these sediments into the lab and feed the microbes in there with, for example, ibuprofen or metformin. One is a drug that you might know from acting against headache. The other one is used for treating diabetes. And here we want to get bacteria that feed on these compounds. That is, they use it as a carbon and energy source and can grow on it without the need for any additional carbon-containing nutrient.
Okay, that sounds very, very interesting. So thank you very much, Marcus, and good luck with the research.
Thanks for the interview.